Biomarkers have the ability to help you improve cost efficiency, predict and minimize risk, avoid late-stage attrition, and make better, more informed decisions. BIOMARKERcenter from Thomson Reuters, the world authority on the pharmaceutical industry, is a single, comprehensive resource, providing you with the information you need on biomarkers at all stages of development.
Good morning, everyone. Beautiful spring morning, isn't it, here in New Jersey? I'm a pathologist. I think some of you know who pathologists are, but I'm a little bit different than the typical one, and I heard a great story once about how you tell an introverted from an extroverted pathologist. When you meet an introverted pathologist in the hallways in a hospital or research lab, a pathologist kind of looks down at his feet when he's talking with you, but if you meet an extroverted pathologist, he'll look at your feet when you're talking with him. So I'm going to hope to be a little more extroverted than that and talk a little bit now for the next 15 minutes briefly about an introduction to biomarkers and their role in drug discovery and development. Way back now, some 58 years ago, George Merck was already talking about the idea of getting the development of pharmaceutical agents directed towards individual patients rather than to groups of patients, and that the medicines that were being developed were for the people, not for the profits. And when you keep that in mind, the profits always follow. They never fail to appear. That kind of sometimes we think of as the earliest dawn of the era of personalized medicine. Personalized medicine is not driven only by science, that's for sure. If we look at the slide and see the various different factors that are influencing it, we see that a lot of it is in the consumers, the customers, the patients themselves, being driven by the issues of how healthcare is financed, how much of their healthcare comes out of their pocket, even if they're insured, their availability to information via the internet and broadband connections, and their knowledge through the nightly news, the internet, and all the types of hallway conversations they have at work. It seems like every day there's another genetics or genomics-based discovery that's made the front page of most of the newspapers in the United States and worldwide. Every day this happens. Today I noticed the genomics headlines of the Today are this diversity of diseases seemingly being related to similar genetic pathways. Most interestingly today is the discussion that there's a lot of similarity between muscular dystrophy and adult coronary artery heart disease. They have genes involved in both, so why not start trying to use the drugs we developed for coronary disease to treat a disease, muscular dystrophy, that currently has no medication that works. Complete new thinking. I think we'd have to say we're all out of the box now, not just a few of us. And that includes the pharmaceutical industry itself. Clearly, some 20 years ago, it was still a paradigm that drugs should be developed for the entire population of a disease group and not in any way developed for a subset or a segmented market of them. But as we'll see, particularly in the second half of my presentation, which will occur in the scientific breakout session, now the development of drugs specifically linked to diagnostic tests, segmenting that disease group into more likely to respond patients, is a holy grail. It's now the goal of the development of drugs, and not just for cancer, but for a wide variety of different diseases. Obviously, finance has a big part of that. We look at the data, we see how much more we're spending in R&D in the pharmaceutical industry and how few new entities are being released each year, whether that's in part the lack of discovery of the drugs themselves or the slow pace of the regulatory process. Nonetheless, it costs more and more every year to bring a new drug to market. And the old phenotypically derived drugs are now being described year in and year out as having substantial problems not known at the time they were FDA or EMA approved. We know that the old phenotypically described drugs cause hundreds of thousands of patient adverse effects in the US, that hospitalizations in excess of 2 million occur simply due to adverse drug reactions. It's a top 10 cause of death in the United States, and it has at least a $100 billion financial impact. It's been theorized more than 10 years ago that 20 to 40% of patients today are actually on the wrong medication because it was not described and found specifically for them. And here's a history. I think this is kind of the rogues gallery chart of agents that one would not want to read about that made it to market and then were recalled for a variety of unexpected adverse events, whether it's the muscle severity of muscle complications of the Bakol type of statin, all the way through, of course, to the celebrated story with Vioxx. Clearly, how many other drugs are on the market now that wouldn't be approved if they were represented to the FDA today? It's a sobering question to ask. So into the space comes the concept of biomarker. And one of the things those of us who are interested in biomarker research always like to do is not to make people feel this is just a repackaging of old words, nothing more than semantics, rather than a complete revolution in thinking on how drugs should be discovered, developed, and ultimately used for patients. So here's my very long sentence definition of a biomarker, a little bit more detailed. A series of gene sequences and mutations, messenger RNA expression profiles, tissue proteins, and blood-based tests that can be used to detect the predisposition for disease, screen for its presence, confirm its diagnosis, assess its severity, predict its response to available therapies, and monitor its clinical course. So as you can see, the term biomarker is not specific for any one point in the drug development cycle, but can actually be applied well before there's even a known need to develop a drug, because the disease is ultimately discovered because of the biomarker research.